Catalytic. And yeah, he is working on amyloids and the history of it. But he will
Can you say the name again, please? To say the name again.
Arunavo Chatterjee.
Thank you.
Hi everyone, myself Dr. Arunavo Chatterjee, and I'm working under Professor Deichmann, and here I am a Deichmann fellow and I'm ma'am. And this is the title of my talk today, the historical development of catalytic amyloid research. So the basically the main topic that I am working on in my postdoctoral project is the catalytic property of some amyloids, specifically the amylin amyloid, which we find the main reason behind the diabetic patients or the type 2 diabetes mellitus. So this is the whole project that I am currently working on in my chemistry lab. But here in the Jacques Loeb Center, my historical or philosophical project actually aims at understanding few of the things or historical development of amyloid research, rather specifically the catalytic amyloid research. So these are the things that I am actually targeting here. The understanding why amyloids are known as misfolded proteins, the first main thing. Then I'll try to reveal the origin of the term amyloid a bit. Then I'll try to demonstrate the early change of concept on the amyloid research. And finally, the three final sections will show the development of the techniques that modern amyloid research is holding on. And so I'll also try to show how amyloids were introduced into pathology and how they became an object of chemical research, specifically on the catalytic residue. So to introduce the amyloids, I'll try to say a little bit about them. They are the abnormal or misfolded form of proteins, which are self-assembled form of the proteins on highly ordered and unbranched fibrous deposits. They are mainly full of cross-beta type structures like that, and they are full of cross-beta structures like that, and they actually are very much important in pathological aspect, because there are almost 50 diseases in the day today, which are almost untreatable, like Alzheimer's, Parkinson's, or type 2 diabetes mellitus. We all know about these things. But there are some functional amyloids also, which not only plays some beneficial roles, but they can also be taken for different applications as well. So as I told, the term misfolded proteins, and also when I discussed with ma'am, she also told me why these are known as misfolded proteins, why they are different from the denatured proteins. So this is the difference. First, if the protein folds in a normal pathway, it will fold in a particular 3D cell, and they will function properly, or they will whatever they are required to function in our body. But in a misfolding manner, or if the function or folding doesn't form or doesn't happen correctly, they can form this type of beta sheet rich structures, which ultimately turns out to be insoluble, and they can show some abnormal conformations as well as abnormal functions upon some certain conditions. So what is the difference then? So it's quite obvious that they have some alternative structures, that the denatured proteins do not have any structure at all. So the denatured proteins completely loses its functionality, and the functional structures also remain unfunctioned or completely disordered. But the misfolded proteins do have some systematic structure, which are these amyloid fibrils or beta sheet rich structures, which we know as amyloid fibrils. So they can have different functions, they can have abnormal functions as well, but this unfolded proteins does not function properly at all. So this is the basic difference between the denatured protein and the misfolded protein which forms the amyloids. So after getting some idea about the amyloids and the formation of them, so I am trying to reveal the origin of the term amyloid. The idea actually stems back to 1639 by Dr. Nicolas Fontanes, who actually unusually found some homogeneous material in the liver or spleen of some tissue during autopsy of a young man who was actually suffering from jaundice. But although the chemical nature and also the detailed structure was not known at that time, but this accounts to be the earliest research of amyloids in pathology. Then came the term amyloid which was coined by Matthias Jacob Leiden in 1839. This was a
first usage of the amyloid term, and he actually, Schleiden was the founder of the cell theory, and he actually wrote in his book, that Principles of Scientific Botany, that this term for the first time, which actually meant something like starch, because he actually used the plant tissues to testify by the iodine sulfuric acid test, which actually is given by the starch when it becomes blue upon iodine sulfuric acid test. Similarly, these plant tissues were also turned into like this amyloid like starch-like color upon introduction of iodine and sulfuric acid. So he termed it in that way, and he told that it could be a starch-like thing or carbohydrate-like thing, but actually it was a misnomer. We will find out. We will find out why. Then came Rudolf Virchow around 1854. He also did some experiments of the abnormal deposits in human organs, and also this time also he showed that iodine sulfuric acid tests did give some blue coloration like the starch. This time he told that deposits were carbohydrate-like. That's why he again used the term amyloid like Schleiden did. But again, this term was not correct. We will see why. So this is the time when the change of the concept came. In about 80 years, it almost took about 80 years, the Frederick Daniel and Frederick August Kekule in 1859 suggested that amyloid may not be carbohydrate. They can be nitrogen containing or proteinaceous, because they saw that the high proportion of nitrogen was found in the organs where infiltration of the amyloids was done. So they assumed that they actually mainly represented albumoid compounds. They do not find anything like cellulose or carbohydrate in those particular substances. So this is the first time when people just went out of the box and thought that these amyloids might not be carbohydrate-like, they can be of nitrogen-containing proteinaceous substances, and this is was supported by several other scientists like Samuel Wills or George Bart later also. So after getting the change in the early concept or early idea of the amyloids, people tried to develop some techniques that could characterize these amyloids through different ways. First of all, that came was this staining method. So at that time there were no microscopy of the things or crystallography what we use in modern times. So this is the first use of the staining dye like methyl violet on 1875 by Richard Hessel, Rudolf Jürgens and Andre Victor, who actually used the methyl violet for detecting the amyloid, and they discarded the earlier used iodine sulfuric acid test, which actually gave the color of starch. And then also in 1876, Soika Jeprak reported having found amyloid in the cardiac tissue that was in the live cell by using the same method. Actually then William Ackroyd and Paul Ehrlich, they termed this methyl violet as metachromatic around 1878. All these studies was done using the bright field microscopy. Then came the another important dye which is known as Congo red. It was Belgian physician Paul Divry and biochemist Marcel Florquet's discovery in 1927, when they found that this Congo red can also stain these amyloids and they can give distinct apple green birefringence when they are seen under the polarized optical microscope. In 1922, German chemist Hermann Benhold further discovered what was the exact interaction between the Congo red and the amyloids happening inside the inside the tissues and also outside the tissues as well. Then came another revolution, I should say, in the field of amyloid research, the discovery of fluorescence microscopy. Or rather the introduction of fluorescence microscopy in the amyloid research. And it happened in 1959 when Dr. Philippe S. Vacher and Charles F. Ekuling used the microscopy for the first time to detect the amyloid, and they wrote their all these observations in this book or rather the seminal paper, and they discovered this dye, benzothiazolyl dye, thiaflavin T, which is actually used for us also, by us also in that amyloid detection in our lab as well. Then came the dye thiaflavin S in 1962, which is another derivative of this thiaflavin T, when Gabor Kellini, a prominent Hungarian pathologist, used it for imaging the senile plaques and neurofibrillary tangles in Alzheimer's disease. Dr. Holt Puchler, a highly influential German-born histochemist, also used this Congo rate as the fluorescent level, which was earlier used for the birefringent or the polarized optical microscopy of the amyloid, was used for the fluorescent model as well. But all these studies again use the bright field microscopy. This exploration is under process, and I have not finished this exploration yet. So the historical development of other techniques and also fluorescence microscopy is going to be done in the due course. But now I'll jump to another thing that was very important as well, the introduction of amyloids in pathology. So after discovery of Nicholas Fontanes, I hope you would remember that in the first slide it was Nicholas Fontanes who used the term, or rather discovered the amyloidaceous substances in liver or spleen. After that, almost 2 centuries went past, and in 1901, Alzheimer's disease was first identified clinically by German psychiatrist Alois Alzheimer when he examined August Deter, who had been suffering from early memory and language problems. So this was the first detection of Alzheimer's, and in 1906, Alzheimer's, he actually wrote this paper and published it and reported this thing. But in 1910, German psychiatrist Emil Kraepelin, who was happened to be his colleague, actually coined the term Alzheimer's in memory of his colleague. But it took again almost 85 years in 1984 when American research pathologist George Glenner found that the beta amyloid peptide is the true culprit of the Alzheimer's disease which is happening in our body. Then came another important pathological discovery of the pancreatic islets of diabetic patients. Dr. Niels Gallerset in 1938 proposed is that para-amyloids at that time, it was actually the islet amyloid polypeptide commonly known as amylin, which they found to be amyloidaceous in the pancreatic beta cells of diabetic patients. This actually was the first introduction of the amylin polypeptide, which was later characterized after almost 40 years in the later 1980s. I beg your pardon, this part is also under construction because the pathological impact is still a term which I am working on. And the chemical, another important aspect that I already started my talk with the title that historical development of catalytic amyloids. But not, if I see that the role of the amyloids in the pathology, we might see they are not that much beneficial, they are detrimental, but they are not actually. There are some scopes and people have developed the object of chemical research specifically as a catalytic residue in future. So if you see this classification, you can have an idea. Now the amyloids are classified like this. These are the pathological amyloids I already talked about. The functional amyloids can be like structural, they can form different structures and do different functions. They can be of storage, applications. These are the ones that I am interested about, the catalytic amyloids, and we can also synthesize or engineer amyloids depending upon the sequence of the polypeptides. So even the catalytic amyloids can be categorized into categories. Naturally occurring catalytic amyloids, like the pathological ones, like we are already having the amyloid beta amylin and alpha synuclein, for example, in our the persons who suffer from Parkinson's, Alzheimer's, and diabetes. We can also channelize or engineer catalytic amyloids by sequencing different peptides in different cell assemblies, and they can show catalytic activities like esterase-like activity, peroxidase-like, ATPase-like, and oxidase-like activities as well. So what is the main reason behind their activity? They have an ordered, repetitive arrangement of catalytic residues, and the crowding effect or the molecular crowding effect is also a very important factor when they are showing these catalytic activities. The large reactive surface actually in these amyloids is the main reason behind the substrate facile substrate binding, so that they can perform the catalytic activity feasibly. Although the pathological impact and all other discoveries on amyloids was stemmed back to 1639 or even the 1700s or 1800s era, but the catalytic research did start very recently. On 2014, it was from Dr. William F. Degrado and Ivan Koryendovich group, which they synthesized this small 7 residue amyloid forming peptides and showed that they can show ester hydrolysis. This was actually the first report for the amyloids who can act as a catalytic residue. This observation supports the potential link between amyloid assemblies and the emergence of protein catalysts during the evolution of enzymes. This part is also under construction, so few of them I am still studying on and trying to develop them for my study. So if to summarize all the things, we can say that earlier coined by Sliden, the amyloids were found to be of high nitrogen content by Mr. Frederick August Kekule. They actually went beyond the idea given by Rudolf Virchow that these amyloids are carbohydrate-like. Those are eventually not. And still, although we use the name amyloid was, sorry.
Oh, no.
The work by Richard Hessel also used methyl violate for the first time to stain the amyloids going beyond the iodine sulfuric acid test, and the utilization of several techniques like polarized light microscopy, which I already mentioned. Bright field fluorescence can be of hugely important factor for the research in the amyloids, but the introduction of crystallography and electron microscopy, which I have not discussed yet or even gone for, is yet to be explored, and it can be shown that they are the main instrumental factor in the development of modern day amyloid catalytic research. And it was Alzheimer, Dr. Alois Alzheimer, who actually reported amyloids in pathology after the 17th century, who actually reported the first case of Alzheimer's disease. And 2014 was the time when catalytic amyloids was first reported. So it will be not an understatement to mention that the exploration of amyloid research was driven by the development of various techniques like fluorescence microscopy, X-ray crystallography, and electron microscopy, and also by the recognition of the fact that they played important roles in human pathology and also in the functional diversity in different timelines of different years. So there are some open questions yet in my work, that what is the exact pathological impacts of amyloid depending upon its historical development. I'm yet to deliver something about complete historical development of techniques that enable model amyloid research, and I'm also yet to deliver something detailed on the historical developments of catalytic amyloid research. So I was at the end of my talk, so I hectic again.
Yeah, thank you. Time for one question.
Yeah, sure. Yeah, sure.
Okay.
Yeah.
No, I have a very short question. So when was it recognized that amyloids are proteins?
It was back in, I think, by Kekule.
Already Kekule did it.
Yeah.
Yeah, that would make sense. And but why is the term amyloid kept? Because amyloid means in Latin starch.
Yeah, I don't know that, but they kept the term starch-like still in use, although amyloid is not starch-like.
Yeah,
probably because of out of respect to Dr. Sliden and who actually named it at that time, and also Rudolf Virchow, who actually named it at that time.
Okay, thank you. Well,
A little specific question. There's a more relatively recent theory that some of the amyloids in pathological cases can have prion-like characteristics when they move from one cell to one cell.
Yeah.
So I wonder, maybe how or why it was discovered just now. Maybe is it because of rise of computational methods, or was it from the chemical point, or was it from the biological, pathological researchers?
Actually, you're right that the rise of computational chemistry was an important factor in the development of amyloid research recently. So, in tandem with electron microscopy and X-ray crystallography, the techniques I did not talk about here, the computational chemistry was a huge important factor in this case. So I guess whatever you rate is probably some studies were already made or they are publishing the studies only based upon the computational aspect, only computational chemistry aspect. So depending upon the experimental and those data, the the collaboration can finally lead to those type of or answer those type of questions.
Okay. No more questions? Also not by the collaborators? Yeah, all very happy. Okay, then I thank you very much. And perhaps I should say this is pioneering work of us. We never had fellows and we never have done these presentations, of course. And so I thank you very much for participating in that experiment. And I hope it will lead to the benefit of your work. Okay, thank you very much.
Ute?
Yeah,
Yeah, we are on.
Yeah.
Ute?
Yeah.